Good evening, and welcome to our program, Conversations about Landscape Shifting Spring. My name is Mary Miller, and I am the Director of Environmental Science Partnerships at the Exploratorium in San Francisco. We are recording and streaming the program tonight on YouTube and Facebook, and I welcome all of our friends in the Bay Area and beyond. It would be great if you let us know in the chat where you're coming from and whether you've been to one of our programs before or if this is the first time. So great to see some of you are watching us for the first time. Welcome. I'd like to give a short introduction about the Exploratorium and our Conversations about Landscape series. The Exploratorium is a museum of science, art, and human perception located on the San Francisco waterfront. The center of our environmental science programs are in the Bay Observatory, a windowed gallery at Pier 15 with stunning views of San Francisco Bay and downtown San Francisco. We are equipped with exhibits, artworks, and programs exploring the built and natural environment and the ways we can decipher our human impacts. Our work has centered on climate change and the regional efforts underway to mitigate and adapt to the climate realities we face. Over the past few years, the Conversations about Landscape series has helped us generate relationships and discuss ideas like we're exploring tonight. We bring together a community of people with different perspectives, land, people, and place. Tonight's program is also part of a new initiative at the Exploratorium on plants. In 2023, we will be opening an exhibition that explores the science of plants and the many ways they intersect with our lives. We will be presenting ongoing programs like the one tonight to expand on ideas from the exhibition, introduce you to scientists, community members, and artists we are working with. This program and the plants exhibition are generously funded by the Gordon and Betty Moore Foundation, and we thank them for their support. All right, on to tonight's topic, shifting spring. I don't know about you, but out on my walks lately, I've been seeing the signs of spring with flowering fruit trees, and I've been sneezing a lot from seasonal allergies. As we started thinking about the meaning of spring, the Exploratorium team was inspired by a chapter in the Tracking Climate Change at Home by Mark L. Heinlein. He writes spring to encourages his readers to be better observers of the natural world, even if it's just the neighborhood squirrels and birds. Through these observations, he writes we should pay attention to things that are changing about the weather and seasons and take note of how plants and animals are responding to those changes. That's what we're here tonight to discuss and explore, how the seasons are changing, especially the one we're in now, spring. Now, spring means many things to many people, as Mark points out. Your personal science can be to where you live, your observations and connections to your community, and of course, calendar events like the spring equinox or even spring break. Please share in the chat if you'd like what your personal signs of spring are. For instance, when I was a little girl, it was getting a matching Easter dress and shoes. That's when I knew it was spring. Tonight, in honor of the topic and Women's History Month, we're very pleased to have 3 women scientists share their research and insight into the meaning of spring from different vantage points: a woodland habitat in Sonoma County, the microscopic plankton populations in San Francisco Bay, and a community science project that bridges many different ecosystems in Northern California and beyond. Each speaker will give a presentation and we'll come back together for discussion afterwards and look forward to answering as many of your questions out there as we can. Our first speaker is Dr. Lisa McKelly, President and CEO of Pepperwood Foundation in Sonoma County. A hydrologist by training, Lisa directs the research and preservation of a 3,200-acre living laboratory 70 miles north of San Francisco in the Mayacamas Range. Lisa, welcome to the program.
Mary, thank you so much. I'm really delighted to be here. I am so delighted that we're having a Miracle March rainstorm right now. So my talk really is about how we are observing spring at Pepperwood. I'm going to explain what Pepperwood is and talk a little bit about the lens I look through as a hydrologist, and also represent a lot of my colleagues' work who are amazing plant scientists. So I'm really a generalist right now. My passion is bringing together incredible teams who look at climate and ecosystems through multidisciplinary lenses. If you haven't heard of Pepperwood, you're not alone. But we'd love for you to come visit us. We are based in Sonoma County in the Mayacamas Mountains, in between Napa and Sonoma counties. And this aerial photo is showing our Dwight Center for Conservation Science. Actually, this is about November of 2017, after a fire started near that Mount St. Helena in Calistoga and came through our area. So I'm going to talk about spring today. I'm going to talk a little bit about our fire science and how we're bringing these pieces together to understand how climate is changing our environment and what we can do about it as nature stewards. So if Pepperwood's mission is to really work with scientists and decision makers and frontline communities, because right now these issues of how we adapt to a shifting spring are very important in terms of our resilience as human communities as well. So I'm just going to introduce you to the concept of the Sentinel Site at Pepperwood. And I do have to say I kind of hijacked this term from the NOAA Sentinel Site program. The Exploratorium has an official Sentinel Site on the bay looking at changes in terms of climate on the bay ecosystem. And we're looking inland at the mountains that drain into San Francisco Bay. And so I like to think of this as sort of a watershed Sentinel Site. And I'm biased because I do consider myself to be sort of a watershed scientist. And to be clear, I'm the physical scientist who works with all these ecologists. And all these little figures on the screen are showing you all the different places that we're taking measurements on a consistent and ongoing basis to understand how the climate is impacting our incredible biodiverse area. So tonight I'm going to talk a little bit about definitions of spring, which was definitely the challenge that we were given tonight to think about. And I'm going to customize the definition of spring to life on land in a Mediterranean type ecosystem. I'll explain what I mean in a second. And then what are the things we're measuring at Pepperwood, and what are the things you can be observing? What can you contribute as a community scientist to link climate, plants, and I'll talk a lot about the water cycle, because that is the lens at which I look at the landscape through. And then I'm going to talk about climate change. Pepperwood is the hub for a group called the Terrestrial Biodiversity Climate Change Collaborative with the University of California. So we're really trying to, we've done a lot of work sort of projecting climate, thinking about the implications, but we're really dealing with the climate change right now, as you'll learn from my presentation. So first of all, as the physical scientist, I got to refresh my memory about, why astronomy matters and what the fundamental sort of textbook definition of spring is. It has to do with the calendar, the equinoxes, and the solstices. And technically the spring, especially, and this is a very Western hemisphere biased point of view, it's spring equinox to summer solstice. And just a brief refresher, the orientation of the Earth to the sun is what creates a differential in terms of the heat distribution across the planet. So first of all, the equator gets a lot more sun because it's pointing right at it compared to the poles. But the thing that creates the seasons and actually generates the circulation in our atmosphere and our oceans is the fact that there's a tilt in the Earth's axis. It's not totally perpendicular to the sun. And it creates a wobble that creates the variability all over the surface of the planet, without which we probably wouldn't have the ecosystems we enjoy today. So I've got a little video. We'll see if it'll work. So this is showing you the impact of that wobble. And this is really what creates the differences between the northern and the southern hemispheres, and why when we have winter in California, they're having summer in Chile and vice versa. So the kind of ecosystem we live here in the Bay Area is a Mediterranean type ecosystem. So obviously the original Mediterranean, I like to say if you want to know if you're in a Mediterranean type ecosystem, see if you can grow olives or grapes. And since that's some of my favorite ingredients for food, I don't mind hanging out in these areas. But the thing about a Mediterranean ecosystem is that we have a wet winter season that's cool, and then we have a very long, dry summer season. So I'm showing you just Santa Rosa, just sort of the average. This is called a climograph, and this is every month of the year. And you're seeing how the rain builds up. Well, you can see how the temperature is rising over the summer. And I'll show you a hydrologist calendar in a second, because we like to have all the rain in the middle. That's what we're interested in. So we actually look at a calendar that goes from October 1st to September 30th. But I'll also show you that my friends down in Santiago are flip-flopped, but they have the same thing where when it's cooling off, you get your rain, and then when it warms up, you don't have any rain. And just to say, this is different than they have in Omaha, Nebraska, which is a temperate climate where they get most of their rain during the summers, or say in a tropical climate where you're getting variable rain but very consistent temperatures. So this sets up the seasonality of a Mediterranean climate, which is very distinct and can be found all over the world. And they tend to be in latitudes between about 35 and 45 degrees, again, because of the orientation of the Earth to the sun and how it orbits. But I like this ecological definition of spring, and this is one we're going to get into. It's the season after winter, before summer, in which the vegetation begins to appear. And this definition acknowledges that the northern hemisphere is the opposite of the southern hemisphere. So it starts to get into that diversity that we have. But when I started looking at definitions of spring, I actually like the verbs rather than the nouns. To move or jump suddenly or rapidly upward or forward, to originate or arise from the earth. And that's where plants are so exciting. So I have to say, I love the study of water. I started in oceanography. I ended up being a watershed scientist. I worked a lot on fish. I think they're amazing. And when I got to Pepperwood, I had to appreciate plants because that is what is the core of our biodiversity. And I work with a lot of plant biologists, so I'm learning a lot about plants, and they are amazing creatures. So I just want to show you, this is an example of a rainy season. This is after the fire, actually. So 2017, this whole area has burned. And this is showing you the cycle where it's raining, it's kind of cold, but the plants are getting established. And then here comes the spring temperatures, right? It's in March, it's starting to warm up. And so the plants are like saving up all their energy, and then boom, it's like an explosion or a firework of biomass that comes up and out of the ground. And now things are starting to dry out. You can see the seeds forming, and the plants, well, they go to sleep. It's called senescence. So those weren't the most handsome plants on the preserve, so I have to show you some really stellar beings. But this is an example of what the spring looks like in our grasslands. This is one of our ecologists laying a 50-meter long tape. They're flagging along the way because every 1 meter along this way, they're going to drop a quadrat. And they measure, I'm not kidding you, every single plant in there, they are going to try to characterize. This is detail-oriented work. Not for me. This is why I study water. It's a lot easier. But so this is how we take a lens, from a research point of view. And these are just some of the kind of amazing species that we see coming up out of the ground. The milkwort, the California poppy. I don't do the Latin words, I just do the common ones. The lupine that we see. These are butter and eggs, California pink. These are actually fire following species. So we wanted to make sure to put a little pitch in for these guys that they really don't emerge very often unless there are post-fire conditions. And they're in this incredible burst of biodiversity that's been waiting and lurking in the seedbed. And then as a hydrologist, I have to say the other thing about spring is that's when we get our spring runoff. So up in the headwaters, the Russian River, where we're based, these streams are dry most of the time. And yet we have incredible aquatic and riparian ecosystems that rely on this spring runoff that happens, including this handsome salamander, our Pacific western pond turtle. My favorite, the newts. We have all 3 of the California newts of pepperwood, and they're so adorable when they're this size, and then we want them to make it to here, to be chunky newts. And then a whole nother conversation could be about our wildlife and how they sync up. But I'll just say that every single one of these species we just saw has some sort of life cycle that's driven by the spring and stimulates them. So we can introduce the concept of phenology. Phenology is the study of the timing of life cycles. And I just want to say this is not a new science. It may be the most ancient science that we have. This is the Yawuru cat calendar, with apologies to the Yawuru people here. This is a First Nations tribe in Australia, and it's showing you that they had a very in-depth understanding of how the different seasonal cycles that are in the different colors link up with the animals and the plants that they rely on for their well-being. You think about it, that people did not have a digital clock, but they knew what time it was based on what was happening with the ecosystem and what the relationships were, in particular between the life cycles of the different animals to each other, because they understood the food chain and the relationship of these connections. So I'm going to talk a little bit about our work doing plant phenology, which is studying the life cycle of plants. And this is Gwen Twomey, who's one of our phenology volunteers and stewards. And we have been collecting now since 2014 very detailed information. You can see Gwen's holding her data sheet on plants. And our volunteers go out 2 times a week to make these microscopic observations, literally with magnifying glasses, to see what these plants are doing. So what are they looking for? They're looking for major life changes in the plants. And so I can't help, because I'm learning about plants, to sort of relate it to our own life cycles. Leaf break, right? You have a totally bare twig, and then you see these little green shoots growing out. Birth and growth, and the plant is putting on leaves to be able to photosynthesize. Flowering, showing off, trying to attract a mate or perhaps a bee. Reproduction is happening in the season. And then fruiting, which to me is like, okay, you got to get those kids to college, get them landed, and then you can chill out. So what they're looking for in the field, over my very gross oversimplification, are these clues about what phase the plant is in. So they're looking very closely at the buds, the new leaf growth, the flowering elements that come out on the plant. And one thing to keep in mind here is that the plants and the insect life have really evolved together in terms of the spring cycle. So one of the concerns we have about climate change is what happens if the food and the pollen gets out of sync with the pollinators. Big question, I'm sure we'll talk more about. And then the fruits that get put out and prepare for the plant to, go into a quieter cycle when they have less water to support them. So this is just an example of the oaks that we're monitoring. Each of these labeled oaks is an intimate friend of one of our plant detectives. They go out twice a week, and they're looking specifically at those oaks. Since 2014, we got this number this morning, 117,817 observations made of these plants. This is a good job for those detail-oriented people. And then in the bottom, we're sort of showing you the life cycle that we might see, for example, at a black oak, which is actually one of our most culturally significant oaks on the preserve. So I just want to introduce one sort of mathy thing, well, maybe 2, but growing degree days is one of the metrics that we collect with the National Phenology Network, and it's what allows you to compare warmer and cooler years to each other and really see the impact. What we call, when we talk about a shifting spring, the growing degree day is sort of adding up all the warmth or adding up all the energy that you might have to be able to grow and do your thing as a plant. And so you pretty much assume that they're not going to be very busy until you start to get days where the temperature's above 50 degrees, which is just about the weather we're getting into now. And it's pretty easy math, but you deduct the space temperature 50 degrees from whatever the maximum temperature is of the day, and you add that up, and you get this degree day number. So this is the days of the year since January 1st. So we're only getting about a third into the year, the first 4 months. And this is an example of a warmer year because you have more growing degree days earlier in the year. This is a cooler year because you're seeing less, and you can see the offset between the leaf break, the flowering, and the fruiting. And it can be up to about 20 plus days difference based on the accumulated heat that these plants can collect. We contribute our data to the National Phenology Network because it allows them to aggregate these results across the entire country and compare it to historic conditions. This is what they call the Spring Index leaf anomaly. But what it's showing you on this map is how much earlier is spring coming to this area than in what we assume is a pre-global warming scenario. And we're already getting spring like 10 to 20 days earlier. That's up to 3 weeks earlier because of global warming. And I just want to put in a pitch, like this is an opportunity for anyone who's this detail oriented and loves using magnifying glasses, like go to the National Phenology Network website and sign up. You can start collecting observations. So this is an example of the kind of projection work that we do at Pepperwood with our partners. And we're looking at the historical trends in gray and then the potential future trends in growing degree days. So we've already seen a pretty significant increase, although this graph is not that steep going up. But in the future, we're really looking at up to a 5+ fold increase in degree days at the same point in time, right? So big questions about how will that affect our plants? How will our plants will adapt? How will the animals that rely on those plants adapt? Just can't help throwing in some details about fire. This is a busy map, but this is our property right here. This is the 101 Santa Rosa. Just showing you what our neighborhood has looked like since 2017. Our entire property burnt in 2017. Half of it burnt again in the Kincaid. Last year, we were helping our neighbors and expecting it to possibly come to us. This is a glass fire. This is an example of how we use remote sensing to look at the variability in fire severity across the area. So fires are coming to our region and resetting the clock on our vegetation to a certain extent. And so we are working with David Ackerley's lab at UC Berkeley and many others to monitor this change and understand, because really these fires are the mechanism of climate change impacts on our vegetation. It doesn't happen gradually. We always knew fire would probably be the most important mechanism, and now we're getting to observe it. So here are our intrepid plant detectives, Eleanor and Wendy. And this was actually the first time that a National Phenology Network transect burned. And so these guys were tenacious enough to figure out how to reestablish it and how to start over and what kind of assumptions would you make. And you might look at this plant and think that it's dead, but it's not, because they're reaching out, and this plant's whole strategy is to resprout from its roots. So this is a fire adapted organism. It's completely adjusted to this fire, and it's going to spring back from that little shoot down there. And so we have been tracking these plants post-fire and establishing how to integrate them into the phenology database. And this is just an example. I'm not going to try to explain it all, but this is the kind of detailed calendar you get about this plant. So you can compare between 2016 and 2021, when did it leaf out? And one of the things you might see right away, for example, after the fire, it ended up having young leaves year round, because that's its recovery strategy from the fire. It's going to put out new leaves the whole time to rebuild its tissues. And there's more to see. We didn't actually get fruiting, for example, flowering or fruiting, until the second year. So the plant is saving its energy for certain life cycles to rebuild. So I'm going to step out of being a talking for the plant ecologist and just say, as a hydrologist, how do I think about the seasons? Because I took that seriously from Susan. And first I thought, well, yeah, for hydrologists, there's kind of two, wet and dry. And I was like, well, okay, it's a little bit more complicated that, because in the wet season, we have the cooler part and then the warming part when we see the plant activity. The plant activity itself takes up water that impacts our water cycle. And there's kind of the dry getting drier, and then there's the dry getting colder parts. So again, as a physical scientist, those are my main drivers. And of course, I had to start saying, now I'm learning about the fire season. And so the fire season is that period when things are so dry, we really have a high risk of ignition if something breaks out. So this spaghetti plot that you're seeing below is a plot of our rainfall measurements, and that's what these spikes are. These show different rainstorms that are happening. And then these spikes that kind of taper down. These are soil moisture probes at multiple depths, and we have these now at 17 different sites at Pepperwood. And we're really fascinated with this cycle of wetting and drying and wetting and drying that happens in the soils. It's feeding the ecosystem. It's a very important reservoir of soil moisture for all the vegetation. And one of the things I want to show with this graph is just how important spring rains can be. And so it's not the total amount of rainfall you get in a year that matters, but the timing really matters. And that's why I'm so happy about the rains we're getting right now, because we were looking at quite a severe drought. We only had 30% of normal rainfall at Pepperwood. And this spring rain is going to help refill these soils and then give us more time for this. Oops, okay, what went backwards? To go down the ski slope of drying out. And this point here, we're just pointing out, this is where the plants get so dry that they pretty much shut down their respiration. They can't operate any further. They got to hang out through this whole period until the rain comes back. And again, this is the October 1 to September 30th calendar that we like to look at, because this is all the interesting part, you guys, like what happens when it rains. So that's the hydrologist view. And I'll just say in terms of climate change, this again is some projection work that we've done with our partners. Here's 2006. I picked this year on purpose because I remember doing field work out on the streams of Sonoma looking for fish habitat and getting rained on in April, May, and June. Not that common, but it happened that year. And it shows you that we had a nice, long, wet season, and we stayed pretty moist through the summer. But here's a projection from 2094. This is not to say we think this will exactly happen 2094, but this is the kind of scenario we could be looking at if we continue on this arid trajectory. And you can see that the wet season is much shorter. We're not getting those spring rains, and so the degree of drought and the degree of fire is a much bigger issue. And really, this is happening because as we add heat to the environment through global warming, it's like a blow dryer hitting the soil, evaporating more of that water into the atmosphere. So it's not available to be part of these spring runoff events. And I should say, we're going through the year here, the lighter blue is runoff and the darker blue is recharge into our aquifers. So that's my take on shifting springs at Pepperwood. Please do check out our website if you're new to us, and there are spring wildflower tours, so you could come be part of our observation team. We're also joining the City Nature Challenge, which Rebecca will be talking about later on in the presentation. Thank you.
Thank you so much, Lisa. Before you go, we had a question that came in about rain and plants. We had the question, one of the most amazing examples of spring I've seen is the super bloom in the Death Valley. Can you talk about why it's rare and the role water and seasons play in some of these more dry adapted places?
Yeah, well, I mean, that is, and I got to be in a super bloom outside of Palm Springs the last time it happened. And that is that perfect timing, right? That is enough water coming in the spring season that the plants have the perfect conditions of where they both have water and heat to do their thing. And so that's what's so interesting about the plant life of California, because it's adapted to extremely variable and in the desert harsh conditions. And yet there are certain plants that will hang out in the seed bed. And I remember seeing a couple barrel cactuses and amazing other flowering cacti and having explained to me, like, these haven't bloomed for 30 plus years. So there are a lot of organisms in the California ecosystems that hang out until their niche happens, until their perfect condition happens. And that's when we get blessed with these surprises. So that's a short way of putting it, but I'm happy to share more about that.
That's so great. Thank you so much, Lisa. And I really took to heart what you said about the timing of the rain is as important as the quantity of the rain. So I'm going to introduce our next speaker. Thank you, Lisa. You'll be back. Our next speaker is Tara Schraga, an oceanographer for the USGS Geologic Survey and manager for a research program that has been tracking environmental conditions and plankton populations in San Francisco Bay since 1969. Tara hasn't been doing it since 1969, but she now manages that program. Tara, thanks for joining us tonight.
Hi there. Well, I'm excited to jump right into it. I'm going to talk a bit today about chasing the spring phytoplankton bloom in San Francisco Bay. And let's see, I have control. I think. Do I? I don't seem to be able to control the screen.
Okay. Sorry, folks. We're just going to try to get some control for Tara.
All right. I have now control, maybe? No. I don't.
Not quite. Well, why don't we go to the first slide and we'll just see if we can figure this out. You can always call for the slides.
Okay, why don't I do that?
Yeah, so if we could just advance to the Tara's first slide. Yep, we'll go to the next slide.
Oh, no, great. I think I might be able to. I think I have control now. Okay, great. All right, so to start out, what are phytoplankton? The word comes from the Greek, phyto for plant, and plankton for wandering or drifting. And so phytoplankton are single-celled microscopic plants that float along in the currents. And you can see I've got photos of them here. They are diverse, different shapes and sizes, and some of them quite beautiful looking. I was hoping that I could go to the next. Okay, I did it. Okay, so why do we care about phytoplankton? Well, they produce half the oxygen of the world through photosynthesis. So the other half is the land plants that Lisa's been talking about. And so you can thank the phytoplankton for every other breath that you take. Also, why we care about phytoplankton is that they're the base of the food chain. They fuel the whole San Francisco Bay food web. They fuel every aquatic food web. So everything either eats a phytoplankton or eats something that ate one. All right, so how do we measure phytoplankton? We can't use a botanist quadrat to look at these microscopic plants. And so we have to find some other way to quantify them. And one thing that phytoplankton have in common with land plants is the green photosynthesizing pigment called chlorophyll. And luckily, chlorophyll is easy to measure. We grab a water sample, we filter it, and we analyze it in the lab. And so we use chlorophyll as a proxy for phytoplankton biomass. And this green pigment, chlorophyll, is one of the ways that we follow the changes in the amount of phytoplankton that are in the water. And chlorophyll is one of the things we measure as part of a larger sampling program in San Francisco Bay. So San Francisco Bay is an estuary. It's a water body that bridges the land and the sea. And San Francisco Bay is between California's two largest rivers, Sacramento and San Joaquin, and the Pacific Ocean. And as Mary said, for over 5 days, the USGS has been maintaining a program of research and observation with the goal of understanding how estuaries function and change in response to the natural forces and human activities. And ours may be the longest continuous sampling program in the United States. So in the middle of the slide is our sampling stations along a map of San Francisco Bay. We sample along the Deepwater Channel in our research vessel monthly with some extra sampling in the spring. I'll get to that later. And in the lower left corner is Dr. Jim Klern. And he has been the director, the chief scientist until his recent retirement of this Bay sampling program. But when he first started, they didn't know anything about San Francisco Bay. They didn't know how this estuary functioned. And in fact, they didn't, like estuaries in general were not yet understood. And so they spent the first years having sampling cruises to look for the spatial and seasonal patterns. Okay, and one thing they discovered while measuring the phytoplankton as chlorophyll was that there's a strong annual pattern of chlorophyll peak values. Okay, so in this plot, I've got the chlorophyll on the y-axis, so that phytoplankton biomass, and then the x-axis, it's every year, and each dot in this plot is a cruise, a time they went out and sampled in the bay for the phytoplankton, and the peaks represent periods of rapid biomass accumulation. Lots of phytoplankton growing and cell dividing. And these times, these peaks are called blooms. And in the South and Central San Francisco Bay, year after year, they saw the blooms were happening in the spring. So hence the term spring bloom. And some years there was a big spring bloom, and some years there was a small spring bloom, but they saw it every year. And as we've talked about, you can observe the spring effect in the land plants, how they just come alive. And something similar happens when the phytoplankton blooms. If you could see microscopically into the water, you'd see it teeming with little cells dividing rapidly. And when the biomass accumulates and persists for 2 to 4 weeks, we call it a bloom. So what are the ingredients for a bloom? The first ingredient is growth. So just like land plants, growth is determined by light, nutrients, and temperature. In the bay, the range of temperatures are great for phytoplankton growth. Warmer temperatures definitely encourage phytoplankton growth, but that's not the trigger for the bloom. And the bay is also a high nutrient place. Nutrients are always available for growth. So the nutrients help maintain the bloom, but they don't play a role in initiating the bloom. It's really about access to sunlight, that sunlight energy, and how much sunlight the phytoplankton are receiving. So just like on land, the amount of light that hits the surface of the water is mitigated by things like clouds, rain, storms, and fog in the bay. And then for the phytoplankton, there's a question of how much light penetrates in the water. And that is impacted by the depth of the water. So the deeper you get, less light penetrates. And also how much sediment is in the water, because the sediment is going to block the light. So phytoplankton will get light energy if there's more sediment. And in a turbid, muddy bay like San Francisco, it's quite a challenge compared to somewhere like, say, Hawaii, where there's going to be a lot more access to light through the water column. But if the phytoplankton can get enough sustained light, they can accumulate biomass and have a bloom. Except the other component on how much biomass can accumulate is grazing or consumption. So zooplankton in the water column, clams, consume the phytoplankton. And so there's going to be less biomass in the water if the rate that they're being consumed is greater than the rate that they're growing. No bloom. But if cell division, if growth is faster than the grazing rate, you can get a bloom. All right. An essential bloom ingredient is something called stratification. So in the ocean and in lakes, as we move from winter to spring and the days are getting longer, the sun warms the ocean surface and it creates this layer of warm water because warm water is lighter than cold water. So the warm water is sitting on top of the cold water, and this stratification, this layering is called stratification, that is set up. And the layers inhibit vertical mixing of the water column. They stabilize the water column. And the phytoplankton that are lucky enough to be held in that upper layer get lots of access to the sunlight that they need to photosynthesize, to grow. So when this stratification is set up, you can get a spring bloom in the ocean or in a lake. And it's kind of neat because temperature is driving the bloom here, just like on land. But San Francisco Bay is a bit different. We still need stratification for a bloom, but it's not driven by temperature. Instead, it's driven by that freshwater inflow. So if you look at the map of California, you see this big brown basin, and that basin is the drainage basin into the watershed that Lisa was talking about, and it funnels all of the snowmelt from the Sierras and the rain flow into rivers that then flow into San Francisco Bay. And river flow is seasonal in California because of our Mediterranean climate. And I'm going to go with, we have 2 seasons in California, wet and dry. So it's dry in summer and fall, and there's not a lot of river flow. But then in winter and spring, we get more rain. We can even have exceptional rainfall, like during El Nino conditions, and even flood events. And so the plot on top is showing inflow from the Sacramento-San Joaquin Delta into San Francisco Bay. And you can see those seasonal peaks. And that fresh water that comes in drives stratification in the bay, because fresh water is lighter than salt water. The salt makes the water heavier. And when that flush of fresh water comes in, the water column can get stabilized. And those top layer phytoplankton can have access to sunlight, and they can bloom. So here's a real world example of data we've collected on our cruises. So when we go out on our research cruises, we have instrumentation. We drop sensors through the water column. And these plots depict salinity, a salinity sensor that was dropped through the water column, and it's collecting samples of how salty the water is. And so on the y-axis, we've got depth in the water, and the top of the plot is the surface, and the bottom of the plot is the bottom of the water column. And then from the left to the right of each plot, we've got freshwater to salty water, a whole range. And so looking at the day we went out in January 1997, January 28th, that was a wet winter and some really nice stratification set up. And you can see that the top layer is pretty fresh. And then there are multiple layers till you get to the bottom, the saltiest ones. This structure persisted and allowed for a pretty big bloom to happen. And you can compare that with a profile collected this January of this year. We had a drier winter, and you can see a very well-mixed water column. It's salty at the surface and it's salty at the bottom. And all right, this plot's not completely fair because it's so well mixed, partially because of what the tides were doing. But you get the idea of what a structured, stratified water column looks like. All right, so part of what we do each year is chase the spring bloom. Spring for phytoplankton, it starts anywhere from early February to late April. We don't know when it's going to happen. We can't predict, but we know that it's going to initiate during a period of slow tidal currents. And this makes sense, right? That if you have strong tidal currents, it would break down the stratification. But if you have mellow currents, they're gentle, then that stratification can set up. So we can't predict the timing or amount of rainfall or freshwater inflow, but we can predict these tidal currents. And so we schedule our sampling for late January through May during the slowest tidal currents in the hopes of capturing the spring bloom. All right, so back to that earlier plot of our chlorophyll peaks over time. So with these decades of observation, we learned that there's a spring bloom every year. We learned why it happens in the spring. And we can understand why we get differences in the size of the spring bloom from year to year. Those wet, rainy El Nino conditions and dry drought conditions produce very different magnitude blooms. And now we have a new spring bloom mystery that we have not solved yet. So in the last decade, the spring bloom seems to be getting smaller. This plot is depicting the magnitude of the spring bloom each year. So again, we have chlorophyll, our proxy for phytoplankton biomass, and there's 1 point for each year, and that is the average February to April chlorophyll. So the magnitude of the spring bloom each year, and you see how it's weakened since 2010. It's really dropped off and stayed down there. And we are trying to tease apart why that's happening, but we don't know yet. But what we do know is that this drop-off has to do with changes in the bay that have either decreased the growth of phytoplankton or increased their consumption, right? Are two controlling factors. And yeah, the bay is in a state of constant change. Sometimes it takes us some time to figure out what is driving these changes, which is why we need to keep these sampling programs going so that we can keep noticing the changes and deepening our understanding of ecosystems. All right, so it's not so easy for the casual observer to notice changes in San Francisco Bay phytoplankton. But next time you're driving over one of our 8 bridges or flying over the bay, pay attention to the color of the water. So it's going to be green or red or brown. And brown means that most of the particles in the water are sediments that are being stirred up. Green means there's not so much mud in the water and you're seeing the green chlorophyll. And when the spring bloom is happening, the bay should get greener and less brown. And when it's red, it's a kind of phytoplankton that has red pigments. And you can compare with the blue of the ocean in this picture. And it's blue because there are less sediments and less phytoplankton in the ocean than in the bay. And I want to acknowledge just everyone in my research group and the many, many people who have contributed to, and the organizations who have contributed to and sustained this long-term data set. And if you want to learn more, you can check out our website. You can play with the data. And I'm just going to make a pitch that we have a new website launching in April. So,
great. Oh, thank you so much, Tara. That was really fascinating. And we do have a question for you before we move on to our last speaker. And that has to do with, you talked about the phytoplankton bloom in San Francisco as kind of being a good thing. It's the base of the food web. But how does that compare to what we've heard about? How is that different from the harmful algal blooms we've heard about that are really bad for ecosystems and marine organisms?
Right. That is an excellent question. So, right, the spring bloom that I've been talking about is generally, knock wood, not a harmful algal bloom. A harmful algal bloom is when species of algae that either produce toxins or cause other kind of harm to biota, where their population will increase greatly. And that often actually happens not in the springtime, just based on their physiology, but it is also a bloom because their population increases, and it has different triggers than the spring bloom, and often not the same magnitude, but it has this great impact or this large impact because of the toxins, or some of them are just harmful and get stuck in like gills of fish, things like that.
Well, thank you so much, Tara. We'll be bringing Tara back up at the end of the program for a general question and answer. So if you have more burning questions about plankton in San Francisco Bay, please enter them in the chat. But finally, last but certainly not least, is Dr. Rebecca Johnson, who is co-director for the Center for Biodiversity and Community at California Academy of Sciences, across town from us. Rebecca works with community scientists using the iNaturalist app to document California biodiversity. She's also an expert on nudibranchs or sea slugs, which really are some of my favorite colorful sea creatures. Welcome, Rebecca.
Hi, Mary. Thanks so much for having me. It's nice to be here. I wish we could be in person at the observatory, but it's nice to be here virtually to talk about how we observe in different ways and sharing those observations to help understand our changing world. So we can bring up my slides, Kale. Awesome. So I am a marine biologist, and so when I think about, and I love my habitat of choice is the intertidal, and when I think about spring, I think about when our low tides, when we can go out in tide pools, shift to the early morning. This is a picture taken of me by Allison Young, my co-director, in one of my favorite places, out in the tide pools in the early morning. And this is one of the kind of things that make me really realize that spring is here when we start going tide pooling in the morning. But I live in San Francisco, and so the other thing that really makes me think that spring is here, or reminds me that spring is here, is our blooming street trees. So particularly magnolias, which San Francisco is famous for, and cherries and flowering cherries. So when you start noticing these in the city, it really makes me realize that things are changing. But I've started to appreciate, I'm a marine biologist, like I said, I've started to appreciate grasslands and meadows a lot more through the work that I do in running community science at the Academy. And so you can't really capture the beauty of a meadow from a picture like this. So I just kind of wanted to zoom in and look at, if you look really closely and pay attention, like the beauty that you can see in a spring blooming meadow. And this is something that I wouldn't have paid that much attention to or noticed that much until I started running the program that I do about 10 years ago. So let me tell you about this program. So at the California Academy of Sciences, Allison and I co-direct the community science department. And we have a few goals, and our goals are to connect people to their local biodiversity, help them see that nature is everywhere, connect people to each other, build community around nature, and collect biodiversity data for science management and conservation. And we want to engage people in and have them learn more about natural history science, the kind of science that we do at the Academy. At the Academy, we have natural history collections, and here's just a smattering of the different collections we keep. And these collections are all records of plants or animals, the actual physical specimen, but it tells us where that plant or animal was found, when it was found, and who found it. It tells us where that thing was in time. And just that data point, just those little bits of information, what it is, where it was, and when it was found, help us understand overall, when these are put all together, where plants and animals are found generally, and how that is changing. Obviously, if we have specimens, we can answer lots more questions. We can sequence the DNA to figure out how these animals and plants are related to each other. We can look at the shape to answer millions of questions, like questions we haven't even thought of. But at its core, it's what's called species occurrence data. And when we were developing our program, we really wanted everyone to be able to share those kinds of data, because really everyone can notice something and share what they found. And we use the app called iNaturalist as the platform for our work. So it's an app and a website. I'm going to tell you a little bit about how to use it, but it's free and it's super, super easy to use. And what it is a place for you to take pictures and share of plants and animals of nature, upload them, learn what they are, and then keep a record of everything that you found. I mean, it's more than that, and it can be many, many different things, but at its core, that's really what it is. It's like a digital nature notebook. So you can see here are some different screens. It just shows you kind of the different parts of the app. You can see, you would take a picture and upload it, and then it keeps a list of every single thing that you found. You can also use the app wherever you are in the world and look at what other people have seen all around you. So I'm going to dig a little deeper into how it works. So if you saw this flower and took a picture, you can see this on the far left, take a picture of this flower and you upload it, You can see your little picture here in the center rectangle. And then you can see those core things I told you that those specimens that we have at the Academy that constitute a species occurrence record have. You can see the date, when it was seen, where it was seen, and you can see this, the person who's using this is me. I would be logged in, so you know that I made this observation. And one of the coolest things about iNaturalist is if you don't actually have to know what it is you're seeing, if you click this "What did you see" link, there's a machine learning algorithm built into the app that will make suggestions for you. And then you can choose which one you want. So I happen to know that this is a seaside daisy, so I would click that. And then you end up with a species occurrence record. And you're saying, I saw this plant at this time. And that's just you saying, I think I saw this plant, right? You're using the machine learning. But one of the most amazing things about iNaturalist is the huge community of people online that help you identify what you found. So once you make that observation, this is what it looks like on the computer screen. You can see I said it was a seaside daisy, and then someone came in here and said, yes, that is a seaside daisy. And then there's some agreement about what I saw. And then that data point that picture becomes a data point, and it's what we call research grade. And you can see here there are a bunch of things that it has to have to become research grade. Like if I was talking about the same things that our specimens have, has to have a date, a location, some kind of evidence, either a photo or sound. People in the community have supported the ID. And it has to be wild to be considered research grade. It can't be like a plant that you planted in that's on your your stoop or your front stairs. And once an observation becomes research grade, you can see here this is comparing a plant specimen from the Academy with a similar observation made on iNaturalist, and they have the same core data. It's evidence of the plant, who found it, where it was found, when it was found, and where it was found. And when we began our program, we really wanted people to understand that all of the scientists that work at the Academy, the first thing they do, we all do, is we're curious and we notice things and we notice how things are different. And everyone can do that. I mean, we have expert taxonomists that can tell by looking at all of the different parts of a plant, for example, what is what. But everyone can notice subtle differences, and everyone can take a picture and share it so experts can help identify it. So on iNaturalist right now, there are about 60 million observations that about 1.5 million people have shared, which is incredible, right? So it's not just in the Bay Area, this is happening all over the world. And those research grade observations are shared with the Global Biodiversity Information Facility, which is the same place that all of our museum specimen data are shared. So observations made on iNaturalist help us understand together with museum specimen data where plants and animals are found in the world and how that's changing. So I've talked a little bit about how you can make observations with iNaturalist, and I tend to, when I use it, it really makes me slow down and observe things more than I would normally. I think about the community and the people that I've met through doing this work online and in person, and what observations they might be interested in, and it helps me notice things in a different way. But one of the really cool things is that all of those observations are shared in an open framework, right? So I can go onto iNaturalist, anyone can, and see a screen like this, which is all of the California poppy observations that have ever been made in California. So you can see a map of where those are found. You can see how many people have observed them, how many people have helped identify them. You can go to the page that tells you about California poppies, and again, you can see these data all put together. You can see all of the observations. You can see here this peak that shows us seasonality. It shows when it's observed most. Most people take pictures of flowers when they're blooming. So you can pretty much see when they bloom. You can see over time. So this is, you can see from this graph actually how much iNaturalist has grown, but you can also see that it really blooms the same time every year, or it's a proxy for blooming time. People take pictures of this plant more often when it's blooming the same time every year. And similarly, here's another flower. This is an amazing Mariposa lily. I wanted to show you one other feature. So these are all the iNaturalist data combined, right? This is not really analyzed or modeled in any way. These are just raw data. So you can see this plant, just like poppies, is most photographed or most observed in April. Same, you can see the seasonal pattern of when it's observed most. But there's also a way on iNaturalist that the community can tag photos and mark if that photo is in bloom, in fruit, in flower, or doesn't have any flowers. And you can see here all these red observations are of the flowers, and the orange are of the fruits, and you can see it's a little later in the year. And so these data aren't perfect, but they get better when people take the time to mark observations. And then if you look at all of the iNaturalist data overall, I just wanted to show you a couple graphs. This is a graph made by my colleague, Scott Laurie, who's one of the co-directors of iNaturalist, just looking at when we see American trout lilies, which is more of an East Coast plant. And you can see here, it's in 2016 and 2017, it was observed most often earlier than it was in 2014 and 2015. So is this a sign, like we were talking about, of an earlier shifting spring, earlier blooming trout lily flowers? And I made a similar graph for a California plant called the shooting star, which is one of my favorites. And it's a little harder to see, but you can see that the green line here is 2020, and it's a little bit earlier, the peak, than the next years. And again, these are just raw data. This isn't really analyzed at all. This is just taking raw numbers. But because all of these data are open, scientists everywhere can use iNaturalist data to help answer their questions. So in 2020, there were over 375 papers that were published using iNaturalist data. And I wanted to talk to you about one. And this is a project on mountain goats. So a little shift from plants. We're going to not talk about plants for a second, but we're going to talk about mountain goats. So some folks that study mountain goats and climate change realized they had a lot of gaps in their data because it's very hard to get all over the kind of the rocky parts of North America where mountain goats live. It's hard to get up there. And so they realized that they could fill in a lot of their data gaps with community collected photos of mountain goats from iNaturalist. And what they did is they looked at the photos on iNaturalist and they came up with a scoring system to score the amount of shed, how much the mountain goats were shedding based on photos. And they were able to publish their research about using community photos to really, for the first time, understand some of the phenology or the timing of shedding and mountain goats from these community collected data. And so for me, I want to talk about a few ways that you can participate in some of our programs. But for me, this is just the mountain goat story is a really great example of, you know, if I was hiking and I saw a mountain goat, I would for sure take a picture of it because I've never seen a mountain goat in the wild, and I would share it and I would be so excited. And it would be for me this moment, like this personal experience with an amazing big mammal. But together, that one data point, together with everyone else's story of observing a mountain goat, helps answer some questions about mountain goat biology that we couldn't understand without everyone sharing part of their experience, and that person also becoming a little more connected to nature by sharing their experience. And to me, that's really the beauty of this work. And we've heard it from Tara and Lisa as well, just this like individual data points brought together to answer these big questions. And the thing I like best about community science is it really is people's personal story, their personal act of sharing, that each data point has a story. And so a couple of the programs that we run that I just wanted to highlight that we hope that people will participate in. One is called the City Nature Challenge, which is a global biodiversity documentation contest / collaboration that happens every April. And this is run in coordination between the California Academy of Sciences and the Los Angeles County Natural History Museum, but with hundreds and hundreds of partners around the world. This will be the 6th year of the City Nature Challenge, and this year over 400 cities and metro areas around the world from 43 countries will be participating. And here's just some pictures from previous years of the City Nature Challenge. And really, it's just people getting out in their cities, in their backyards, where they live, and taking pictures of the plants and animals around them and sharing them. This year, it's most likely we will break the 1 million observations over 4 days mark. And the record for number of participants over that 4 days is 40,000 people, and I'm guessing we'll break that as well. And so to me, again, that's one of the most incredible things about the City Nature Challenge is that 40 to 50,000 people all over the world all noticing nature and sharing their observations over the same 4 days is really incredible. So this year, the City Nature Challenge is from April 30th to May 3rd. And here in the San Francisco Bay Area, it's not just San Francisco, it's the 9 Bay Area counties, so all counties that touch the Bay. And so if you're in any of those counties during this time period, you can make and share observations. And we also will post a list of events that all of our partners in the Bay Area will be hosting. So really quickly, I'm just going to talk really quickly about one other program we run. It's called Snapshot Cal Coast. That happens every summer, where similarly, we work with partners up and down the California coast to make and share observations over 2 weeks of the summer. So we can look at what we see at one time period and compare it year over year. So we can see shifts in ranges and other shifts that because of climate change and other factors. And because of this work, we've also been doing this work for 6 years. We get a really amazing picture of the diversity of our coast that's collected by community scientists everywhere. And we're able to notice changes. And one of the changes that we noticed a few years ago was an increase in this beautiful slug. I can't really have a talk without a nudibranch. So here's a nudibranch for you, Mary. We noticed that this nudibranch was increasing in abundance locally here in the Bay Area. And it turns out that it was increasing in many places, but we were able, with the work of our data scientist, Gio Rapacciolo, to take all of the community science data that we have collected through Snapshot Cal Coast and do some modeling to be able to look and see if we can capture this change that we saw in the abundance of nudibranchs. You can see here, this is just a really big peak. That's when we saw this in increasing numbers. And we think it was a result of a warm water blob that happened off our coast in 2015 and 2016. But unlike the work that Tara and Lisa talked about that has really strict front-end protocols that scientists develop, the quadrats and the plankton sampling and the chlorophyll sampling that Tara talked about, for us, we tell everyone, go out there, make observations, take pictures of what you want, and then we do the modeling and the data, like getting rid of the biases and the other inherent problems with people taking pictures of whatever they want on the back end. And so we did this proof of concept to be able to show that we can use community science data to pick up changes that we knew about. And now we're working with the state of California to use some of these same methods to create an early warning and forecasting system for changes along our coast. So we'd love to have you participate in Snapshot Cal Coast. That's this June from the 11th to the 30th. And soon on the Academy's website, we'll have more information about how to participate. And I just wanted to leave you all thinking about those almost 60 million observations on iNaturalist, and that they all started like this, someone being curious and taking a picture and sharing it and noticing the world around them, whether it's a tiny lichen or a flower that's blooming. And those data help us understand a shifting spring from a scientific perspective, but also from our own personal perspective. So I just want to thank my team and everyone who shares their observations. And this is how you can find me and more information.
Thank you so much, Rebecca. God, that's so fascinating. I love the story about the mountain goat. It was,
it's amazing, right? It's like
really
such a creative
amazing
use of these data. So
yeah,
it's so awesome.
It's perfect. Let's bring all three, I'm the fourth, folks up on up on the screen. And actually, I'd like to kick off because there was something interesting that Tara said about in California, we have 2 seasons. You showed the, Lisa, the Yaruru calendar has 6 seasons, but I've heard California actually has 3 seasons, rain, fog, and fire. So can you answer, like, how many seasons do we really have? Lisa, you want to
Who wants to
take that
start?
one? Yeah.
Well, so I agree that fog is super important. By the way, that's our natural air conditioning. That's the big uncertainty. We're involved in fog studies that link the Bodega Marine Lab and Pepperwood to look at that question, because fog it's new territory for research. Let's see, rain, fog, and fire. I mean, like I said, I sort of have wet and dry, but then there's categories of those too. Basically warmer and cooler versions of those two. And the warmer version of the dry is the fire season. The fog is kind of our, those are the cooler, opportunities during the summer and fall period, and they're super important. Do I think it's its own season? Yeah, I guess it is, but fog is very variable across the Bay Area. And so we actually have a great map of fog that our partner, TBCU partner, Alicia Torregrosa made through the Pacific Coastal Fog Network. We're part of a network of stations. So fog is super important, very close to the coastline, and it does get all the way into Pepperwood. We're sort of the most inland area of fog. We're measuring how much water is coming out of that fog. The fog is very important. But I feel like it's a moderator on the dry season. Sure. And we do know that, like, for example, redwoods get up to 40 or 50% of their summer water out of fog. They're actually perfectly designed to harvest that fog. And it's very important for salmonids as well, because the benefit of fog is it blocks the incoming solar radiation. Then it actually contributes some moisture itself. So I see fog as the one of the safety rings that we have during our drought seasons, that if we have good fog, and that's why it's so important. We don't know whether climate change will create more fog or less fog. It's super important. But I don't know if it's its own season. I mean, it's July to October. That's when we expect it. But we can't guarantee it's coming.
Yeah. I mean, I think one of the, we're a coastal climate, even as you said in Mayacamas. So when we have those really cold ocean temperatures from upwelling, that's when we have the foggy season, and it is, as you said, so important.
And it's a whole Pacific Coast issue. It happens simultaneously. So it's a regional thing that drives local climates.
Yeah. So we have a question for Tara, actually, and it has to do with color in the South Bay and brine shrimp. So I think the question is, does the red color in the South Bay, is that from brine shrimp, or what causes, because you were asking people to observe the color of water, and they want to know what is causing that red color in the South Bay.
I love this question. It's phytoplankton. It's always phytoplankton. No, the red, so those are salt ponds that, so it's high salinity, a lot of salt in them. And it's a species called Deneliella. It's a phytoplankton called Deneliella that is salt loving. So it's hydrophilic. And it's kind of neat, right? From the airplane, you can see, when you fly over those, you can see the bright red of those ponds.
Yeah, it's a beautiful patchwork of colors. When you fly out of San Jose, which I live in San Jose, you just see this just kaleidoscope of colors. It's so great. We have a question for Rebecca. And I think you've kind of answered this, but I'll give you another opportunity to take a really good example of the citizen science data that surprised you. Was it an extremely rare sighting, or what? Just a story about something that was just super interesting.
Yeah, I guess. I mean, I feel like everything that if people are willing to share their observations, that makes every observation awesome. But I think that one of my favorite observations was in the tide pools where we do most of our work, south of San Francisco, a place called Pillar Point, it's a really busy intertidal, right? It's not a marine protected area, but it gets a lot of visitors. It has people, it's a really multi-use intertidal spot. So people are fishing, people are there with their dogs, people are there with their families. And we had a volunteer who found a black abalone at Pillar Point, which is one of our only endangered marine invertebrates. And to see, and we found it now subsequently a couple times, not the same one, but a couple different abalone. And it was really amazing to think that this endangered species that is, really not doing that well, to see it thriving in a place where there are lots of people. And it just kind of helped me think about the resilience of that place, and maybe some of the benefits of multiple uses of the intertidal that maybe need a little bit of harvest. So it was a really interesting finding. And then one of my favorite stories is at the same place, we had a volunteer who was taking a picture of an octopus with her camera, her underwater camera, and the octopus actually grabbed the camera. And so the octopus was holding the camera, and she and Allison, my colleague, were pulling the camera back, and the octopus actually took a selfie while it was holding the camera. And so there's an observation of the octopus made by the octopus, which is just like a fabulous story. And recently, I don't know if you've seen all over social media, there's like an incredible photo that an octopus took, but it took a diver's camera that's like, beautiful frameable photo. This was like a kind of blurry octopus selfie, but it was incredible. But really, it is just this idea that people sharing their observations could be something rare. Like one observation could be something, that hasn't been seen in 30 years, which happens all the time. And we always say, like, you could turn over a log and you see a bunch of pill bugs or roly-polies, and it could either be like a super common introduced European roly-poly, or it could be something that hasn't been seen in 40 years. And that's always true. And that makes every observation pretty exciting.
We have a question. Oh, go ahead, Lisa.
I was going to say on the animals taking selfies for us with our wildlife cameras, it's the bears.
Yes,
because
totally.
Bears are so curious and they love our cameras, and we have a lot of like snout, tongue, and they scratch on the camera. So they trigger the camera and take all these incredible pictures. So that's also one of those surprising and amazing things. And folks who are putting their own wildlife cameras in are finding stuff like that too. So.
So great.
I guess Tara, you probably don't have any selfies of phytoplankton taking pictures of themselves. But Lisa, I have a question for you. And that's at Pepperwood. Do you work with in the indigenous people from the area to try to understand more about the breadth of information you could learn about a landscape, and has that influenced how you think of spring or how you think of the place that you study?
Well, we've had a Native American Advisory Council at Pepperwood almost for a decade now, and that's allowed us to build some pretty deep relationships with both people from local tribes who know that particular area very well, and also people from other tribes across the country who've come to settle in Santa Rosa. With respect to spring, most of the issues that we're working with our TEK experts, and I would say indigenous science experts, is how to restore the kinds of disturbances that we used to have from fire. How do we have healthy fire for our ecosystems and cultural burns, some of which would have happened in spring, some of which would have happened in fall. My indigenous science advisors are understanding that we're in a different ecosystem than the one that their ancestors were managing for, a millennia, 14,000 years. And so we really need to work together to bring their detailed understanding of life cycles together with climate science to think about what do we do now, because we have interrupted indigenous land management practices. So I don't have an easy answer for that specifically about spring, but I think the thing for us to understand is the newer arrivals to this area is that the land was stewarded very heavily by our indigenous stewards, and they would have been using fire and doing a lot more disturbance than we're accustomed to. And that's something I think for people who are environmental practitioners and activists understand, is that stewardship was part of the picture to clear away the previous year's debris to make room for the spring bloom. And I think is how I would talk about it. And that's what fire does. Fire takes out the thatch, it takes out that dead and senesced material from the previous year to make room for the spring bloom. And I guess I feel like what they're trying to teach us is people were an agent of helping to facilitate that spring happening. So
yeah, thank you. I know there was a lot of questions in the chat about fire because there's so much controversy about fire, and it really helps to take a deeper and longer view of that. I think we have time for one more question, Rebecca, and I want to throw this to you. And that's about, how do you encourage people to do, community science when they don't feel that they have anything of value to add, or that their neighborhood or their backyard is just not that interesting to be used as data for a scientist to study?
Yeah, I mean, I think it really starts with realizing that helping people realize that nature is everywhere, and us understanding the plant and animal communities in your backyard, or on the sidewalk plantings, or anywhere, like in a city, in the suburbs, not just in places like Pepperwood, but if it's just kind of slowing down and thinking and documenting little things that you see and saying, like, for me, it might be, and this is especially true when we first were, in lockdown in the pandemic, right? Like thinking like, okay, where could I find nature a block from my house? And really paying attention in different ways, and being still and quiet, and, looking for insects, like finding places that there are water, and just being curious about who lives around you, and that it might not be like the most spectacular, it might not be bears, right? But what lives around you is still nature. And the more we understand really about what lives in people's backyards, will help us make connections for wildlife. And maybe it will help us understand what's there now, and how plants and animals are using urban and suburban areas and areas where people live to help us plan better for nature, to help us understand how areas are being used now, and what we can do to enhance those places for wildlife and for people, and bring more biodiversity. I mean, I think being home for me, I've noticed in my tiny little backyard in San Francisco, now I notice like a hummingbird comes like every day at the same time. And then, you can notice like little changes. And I think that helps connect us to whatever place we live in a really powerful way. And anybody can do that, right? Like anybody can take a picture of what lives near them.
Well, thank you so much, Rebecca. And Lisa and Tara, this has been just a fascinating conversation. Thank you so much for joining the Exploratorium Conversations about Landscape.
Thanks for having us.
Thanks so much for having us.
It's been great.
Thanks, the audience out there. We're saying bye for now.